Today we'll be discussing about auditory pathway. Initially, I will outline the auditory pathway and then we will discuss each step. In this video, I'll be discussing about the inner ear structures, if possible, till the superior olivary nucleus, and the remaining structures in detail I'll be explaining in the upcoming videos, how the auditory signal is processed, how we are able to hear different sound, how music is processed. All this I will explain only in another video. This is a basic video in which I will be outlining the most important parts of the auditory pathway. Let us start from the eighth nerve. So we know that the auditory nerve is the eighth nerve, that is a cochlear nerve. So the auditory pathway starts with the eighth nerve. I will tell you the mnemonic initially itself. When I was studying, I used to use this mnemonic, that is E coli MA, so eighth nerve. Then it goes to C. The C is the cochlear nucleus. So ipsilateral eighth nerve. Ipsilateral cochlear nerve goes into the ipsilateral cochlear nucleus. Then it goes to the ipsilateral superior olivary complex. O is for olivary complex. It is superior olivary complex. At the level of the superior olivary complex, some fibers cross over to the opposite side. Some fibers continue in the same side. So from the next segment, it receives contralateral and ipsilateral fibers. So from the superior olivary complex, it goes into the lateral lemniscus. Ipsilateral and contralateral lateral lemniscus. Then I is for inferior colliculus. Then MA is for M is for medial geniculate body. For your MCQ and all, you can ask medial geniculate body is associated with which pathway. Medial geniculate body is associated with auditory pathway. To remember it, you can use music, medial geniculate body. M you can use medial geniculate body, and then it goes into the auditory cortex. So once more, eighth nerve, cochlear nucleus, superior olivary complex, lateral lemniscus, inferior colliculus, medial geniculate body, and auditory cortex. These 7 structures form the auditory pathway. Coming to how we perceive the sound. Remember your physics lesson, sound is like formed by movement of the particles. So it is an energy formed by movement of particles. There is rarefactions and compressions which happens in cycles. So number of rarefactions and compressions per cycle gives the frequency that we see in hertz. So human ear can hear frequencies from 20 hertz to 20,000 hertz. That is 20 hertz to 20 kilohertz. So an external sound is produced that comes and it enters the external auditory canal. Then it goes and touches the tympanic membrane. So the tympanic membrane vibrates, and then which in turn vibrates the ossicles, malleus, incus, and stapes. The malleus, incus, and stapes helps in the amplification and also in impedance matching. About that, I will be doing in another video. And the stapes footplate is attached to the oval window. The oval window is connected to the scala vestibuli. So the footplate of stapes vibrates the oval window and it vibrates the fluid in the scala vestibuli. So the mechanical energy coming from the air is transmitted into the fluid, and then it is converted into an electrical energy which goes via the auditory nerve into the auditory cortex. That is how we perceive sound. We are hearing every day, we are hearing music, we are talking. This is how complex it is. Something we take as very simple in our life is so much complex. It is happening in everybody's body. This is known as mechano-electrical transduction. Mechanical energy is converted into electrical energy. I will come to the cochlea. Cochlea is the sensory organ for hearing. We know that cochlea is a snail-shaped. Cochlea, the word meaning itself means snail. So it's a snail-shaped structure. It is coiled around a central structure. It is coiled 2 and 2 by 3rd around a central structure known as modiolus. The outer part is the bony structure that is known as osseous labyrinth, and inside that you have the membranes that is known as membranous labyrinth. So imagine an eggshell, and inside the shell you have a structure, osseous labyrinth and a membranous labyrinth. Membranous cochlea consists of 3 canals or 3 scala that is known as the scala vestibuli, scala media, and the scala tympani. The scala media is a central one. You can see that the cochlea is coiled like this. If you take a cut of the cochlea and see inside, there will be 3 canals, 3 chambers. So the central one is the scala media, and you have the scala vestibuli and the scala tympani. Scala media, central one, is also known as a cochlear duct. Our sensory organ for hearing, the organ of Corti, is present in the scala media. So that is also known as cochlear duct. And you know that the scala tympani and scala vestibuli have connections to the middle ear. That is, the oval window is formed in the scala vestibuli, which connects the stapes foot plate to the scala vestibuli, and the round window connects the scala tympani to the middle ear. And the scala vestibuli and the scala tympani merge at the apex at the helicotrema. The apex of the cochlea is known as helicotrema. There the scala tympani and scala vestibuli merge, whereas the scala media ends as a blind sac. Another important thing that you have to remember from the anatomy is the scala tympani and vestibuli contains extracellular fluid or the perilymph, which contains a higher level of sodium concentration, whereas the scala media contains endolymph that contains a higher level of potassium concentration. The difference in the ion concentration between the perilymph and the endolymph and the electrical potential difference helps the cochlea do its work. That is how it generates the electrical signals. Inside the cochlea, we can identify three functional units for hearing. The first one is the organ of Corti that is present in the scalar media over the basilar membrane. It is the sensor. That is the one that does the mechanical transduction. That is the one that identifies the sound. That is a sensor. Then you have the stria vascularis. Stria vascularis can be known as the battery of the cochlea that generates the potentials, the endocochlear potential that is needed for the mechano-electrical transduction. Then you have the spiral ganglions, which are present in the modulus, that is central structure, that carries these impulses from the cochlea to the auditory cortex. So there are the three important functional structures of the cochlea. In brief about the organ of Corti, the organ of Corti contains the inner hair cells and the outer hair cells. The inner hair cells are around 3,500 in number, and inner hair cells are arranged in a single row along the entire scala media, around the entire basilar membrane. The inner hair cells are arranged, and the outer hair cells are arranged in 3 rows, just lateral to the inner hair cells. Inner hair cells are medial, outer hair cells are lateral, and inner hair cells are flask shaped, whereas outer hair cells are cylindrical shaped. And these inner hair cells and outer hair cells have projections known as stereocilia. These stereocilia are highly organized and they have actin filled and they have potassium channels. So any movement or deflection of the stereocilia will open the potassium channel and cause depolarization of the cell, and that is how the sound is conducted. Inner hair cells have 2 or 3 rows of the stereocilia that are free floating, whereas outer hair cells, the stereocilia are arranged in bundles of V, and the tallest stereocilia will attach to the tectorial membrane. So what is tectorial membrane is one that covers the entire organ of Corti. It is an acellular structure. The tectorial membrane is medially attached to the spiral limbus and connects the stereocilia of the outer hair cells, not of the inner hair cells. Also, the stereocilia are graded in height. The stereocilia at the base of the cochlea are smaller, and as it goes to the apex, it becomes taller. So the stereocilia at the apex of the cochlea are twice as tall as the stereocilia at the base of the cochlea. That also helps in identification of the frequency of the sound. This is also known as tonotopic arrangement of the cochlea. So the base of the cochlea will recognize high frequency sound, that is 20 kilohertz sound, and that is the shorter stereocilia. And once it goes to the apex, it recognizes lower frequency sound, that is 20 hertz sounds. There are also other supporting cells other than the outer hair cells and the inner hair cells in the organ of Corti. Each hair cell rests on a supporting phalangeal cell. The supporting phalangeal cell of the outer hair cell is known as Dieter's cell. Then there are cells known as the inner and outer pillar cells. These are present between the outer and inner hair cells that delineate the space between the outer and inner hair cells, forming the tunnel of Corti. Then there are other supporting cells like inner marginal cells and outer marginal cells. Outer marginal cells are also known as Hensen cells. Then there are cells like Claudius cells, Bosner cells, etc. All these are supporting cells that helps the function of the outer and inner hair cells. Continuing the previous sound pathway, it comes to the stapes. Stapes foot plate vibrates, oval window vibrates. So a pressure is transmitted into the perilymph in the scala vestibuli. Because of the non-compressible nature of the perilymph, there is a pressure gradient or difference between the scala tympani and scala vestibuli, which causes movement of the basilar membrane. Once the basilar membrane moves, there is deflection of the stereocilia, and hence there is opening of the potential channel and the sound signal is generated. A traveling wave, and if you take a shawl or dupatta or do like this, it goes as a wave. So a sound wave travels through the entire cochlea like this. And what happens is sound wave peaks and then it drops. Where it peaks depends upon the frequency of the sound wave. That is found out by the tonotopic arrangement of the cochlea depending upon the height of the cilia. So the taller the cilia means it is more flexible, so it can detect lower frequency. With a lower frequency itself, it will move. So it will be detecting lower frequencies. At the basal turn of the cochlea, the stereocilia is smaller, so higher frequency only will be moving it. So it detects the higher frequency. And also the width of the basilar membrane differs from the base to the apex, and also the certain small cellular structures are also different from base to the apex. All this contribute to the cochlear amplifier and the detection of the frequency of the sound. Base of the cochlea 20 kilohertz, apex of the cochlea 20 hertz. And the inner hair cells are the linchpins in hearing. The axons of the inner hair cells are the ones that contribute into the spiral ligament and it continues into the auditory cortex. The outer hair cells are mainly responsible for the amplification of the basilar membrane motion, so that we are able to detect even a lower sound pressure stimuli. As I told previously, spiral ligament is present in the Rosenthal's canal in the modulus. It contains the cell bodies of the neurons. The axons of that project into the cochlear nucleus. Spiral ligament, 90% of the cells are from the type 1 ganglion cells that are from the inner hair cells that are thick and myelinated. The remaining 5%, 4 to 5% are thin and unmyelinated neurons from the type 2 ganglion neurons from the outer hair cells. So inner hair cells contribute maximum to the spiral ligament. Then from the eighth nerve, it goes into the eighth nerve nucleus, that is the cochlear nucleus. Actually, each segment has details data to take class for 15 minutes or 20 minutes. So I'll just briefly outline and then details how each nucleus functions. I'll be taking in another class. Cochlear nucleus has two types, ventral cochlear nucleus and dorsal cochlear nucleus. It can be divided broadly like that. The ventral cochlear nucleus is optimized for coding of the local characteristics of the sound, that is the phase, amplitude, etc. It can identify. And the dorsal cochlear nucleus is centered for multi-sensory integration. From the cochlear nucleus, it comes into the superior olivary complex. Superior olivary complex is located in the brainstem, close to the trapezoid body. And superior olivary complex contains three primary auditory nuclei, that is medial superior olivary nuclei, lateral superior olivary nuclei, and also medial nucleus of the trapezoid body. This helps in identification, localization of the sound. That is, this identifies the time difference and sound pressure difference from both ears. If you're hearing a sound from this side, so this side will hear it fast than from the opposite side. So that time difference, the olivary nucleus will identify and it helps to localize the sound. And also sound pressure level difference from where it is coming from, closer or from farther. So that is known as the interoral time difference and interoral level difference. That is sound pressure level difference it can identify. So the medial superior olivary nucleus is associated with interoral time difference. So binaural comparison and timing of the sound. What is the time at which the sound has reached from both the ears? Whether they reach at the same time or whether there is a time difference between them. That is what the olivary nucleus has. So the temporal disparity helps in the sound localization. And also what is known as cocktail party effect. That is, it helps in identifying multiple sounds. In cocktail party, in a party, 3-4 people are talking to you loudly at the same intensity. It will help to identify which person is telling what. That is done by the superior olivary complex. That is known as spatial marking release. That is, competing auditory signals are coming to you, and this helps to localize the different signal from the spatial orientation of the sound. So who has talked what, you will be able to identify with the help of the integration at the superior olivary complex. I'm telling everything in briefly. Each segment I'll explain in detail later. And the lateral superior olivary complex is mainly related with the sound pressure level. That's interoral sound pressure level. With the help of the shadow of the body, the ear, the location of the head and everything, it will identify from where the sound has come. I will discuss about the functions of the inferior colliculus, the lateral lemniscus, the medial geniculate body, and the auditory cortex at the second video. So, for an exam point of view, you have to know the pathway: eighth nerve, cochlear nucleus, superior olivary nucleus, lateral lemniscus, inferior colliculus, medial geniculate body, and auditory cortex. One more important thing that I have to tell you is there are descending circuits for the auditory pathway. Not only does the signals come from the auditory nerve to the superior olivary complex, there are descending auditory pathways. This helps to moderate the stimulus perceived based on our emotional states, based on our behaviors, based on our habits, etc. This also helps to reduce the intensity of sound that goes into the cochlea. And there's also a central mechanism to reduce the trauma from the noise. If you hear a loud noise, maybe temporarily you won't be able to hear for some time. This inhibition can also come from the central auditory pathway.